Hi, Michelle Glass here. Welcome back to another episode in our Chapter 8, Part 2 conversation. We are leading up to our conversation on meiosis. We have already spent the first half of our chapter talking about the process of mitosis. Both are cell divisions, so they are both involved in reproduction of the organism. We've already talked about asexual reproduction as meaning without sex, and we see that that is essentially cloning. In prokaryotes, we see that they do this through binary fission, and eukaryotes, we see that through the process of mitosis. So that was the whole first half of this chapter. We are still leading up to our discussion of sexual reproduction. This involves the special type of cell division called meiosis. Sexual reproduction is important because you're going to increase the genetic diversity of the offspring, because you're fusing together an egg cell and a sperm cell from two different individuals, meaning two different gene pools. We're going to see that those egg and sperm cells are called gametes. And there are different types of life cycles, whether you're an animal or a plant or fungus. All of these are discussed in a little bit more detail in Biology 2. And for this presentation, we're just going to look at the animal life cycle, which is all that's presented for us here in Chapter 8. Sexual reproduction is significant because it's a fusion of gametes, egg and sperm cell coming together. So you have individuals from two different gene pools halving their DNA and then putting that together to make a whole cell, which has given us a lot of genetic diversity in our offspring, which is important for natural selection. Natural selection, remember, is where the environment is selecting for those individuals that are best adapted to their environment, meaning they have their highest chance of surviving and passing on their genetics to their offspring. So the only way you can kind of select for good characteristics is if you have a variety of characteristics to choose from. And remember, environments can change. And so it's important that we have diversity in the population so that when those environments do change, we still have individuals that we can pick out as being good and well adapted for that particular new environmental condition. Sexual reproduction, is producing diversity in three ways. So we have crossover, which is giving us diversity in our gametes. We have independent orientation, which is giving us diversity in our gametes. And then we have what's described as random fertilization, which is giving us diversity in our offspring, or our zygote, which is the fertilized egg. Random fertilization is talking about, okay, the female can make all these different variety of possible egg combinations, and the male can make this whole variety of different sperm combinations. So you have diversity happening in each parent, but then we don't know which one of these special sperm gets to fertilize this egg that was produced. And so you get diversity in the combination of that sperm and egg together. Let's take a look at our animal life cycle. We know this because we are animals and we already have some information about, what it means to reproduce. In the case of the animal life cycle, we have the formation of the gametes happening through the process of meiosis. So that's still the type of cell division we haven't fully talked about. Females are going to do meiosis in ovaries, for mammals in order to produce an egg cell. And then, and let me just go back and say, it's not just mammals that have ovaries. So we see this terminology used really across the board for animals. And then the males are going to be producing the sperm cell through meiosis. So each of these gametes is going to be haploid, meaning it has half of a set of genetic material. When those gametes come together, we describe that as fertilization. The fertilization of an egg is now called a zygote, and that's where we see a diploid cell. Diploid meaning 2N, it has a complete set of homologous chromosomes. That diploid zygote is then going to go through the process of mitosis, producing all of the different cells of the body that are required. So whether we're talking about a single-celled organism or something complex like a human, all of those cells are coming as a result of mitosis. So we see mitosis is for the purpose of growth and repair, whereas meiosis is for the production of those gametes. We said that crossover is one of our important sources of genetic diversity. Crossover is when you have pieces from non-sister chromatids of a homologous pair exchanging information. This is going to happen during prophase 1 of meiosis 1, so we'll be talking about that more in the future. So I have my same set of my homologous pairs here, and I'm showing my sister chromatids, so you can see that the sister chromatids are identical to each other, but the blue chromosome is different from the pink chromosome. When you have crossover, you're exchanging bits. So now we have what we can call recombinant DNA. We have recombined information from the parents, so that instead of just having our original combination of brown eye with hitchhiker's thumb and blue eye with straight thumb, we now have brown eye with straight thumb and blue eye with hitchhiker's thumb. So we've increased our diversity. This individual can make 4 different sperm cells with these different combinations. And keep in mind, this is happening in humans with all 22 pair of chromosomes, all except for those sex chromosomes. The second source of genetic variation is coming from independent orientation. So when those homologous pairs line up during metaphase 1, they can line up in any orientation. So it's not like all of dads go on one side and all of moms go on the other. It's not that it has to have a pattern of, mom and then dad and mom and then dad. So there's a lot of different variations of how that can line up. And so that's going to give us them sorting into different gametes during meiosis. Okay, that's it for now.